Hi, my name is D McKay. I'm a researcher from Australia and I work at the International Agency for Research on Cancer, which is in Lyon in France. I've been involved with Interlymph for maybe 10 to 15 years now, and I'm involved in a number of different things. I'm sitting on the coordinating committee at the moment and also the Hodgkin's lymphoma working group. I'm also involved in the family working group. The family group is involved in trying to understand why certain families have a reoccurrence of certain types of lymphoma. Over the last few years, we've been using approaches that involve the sequencing of the human genome and trying to understand the germline genetic variants that are found in various different patients and understand how those germline genetic variants are involved in the predisposition to lymphoma. One of the most important things that we do inside the working group is we bring together multiple different groups. One of the challenges in this type of thing is it's a rare disease. And so to get enough people together to be able to make for a meaningful study, we need to put together a consortium. And that's what makes something like Interlymph really, really important. Bringing together across international collaboration, a number of different researchers to give us enough people to be able to study these types of disorders. So the project that I've been working with the family working group is based around myeloma. And this is an international consortium that has been put together, and it's based partly on French families. So I work with a national network across France, which has identified a number of different families with a reoccurrence of myeloma in the French population. And we undertook a study inside these pedigrees, these families that have a reoccurrence of multiple myeloma, and identified a really interesting gene that it seems to be linked with the predisposition of multiple myeloma. And one of the biggest challenges we have when we make these types of observations is actually trying to confirm that they are statistically robust and able to be extrapolated to other patient populations. And so one of the strengths in the family working group, but also the consortium in general, is the ability to be able to work with others. For example, here in France, we then worked with a group in the United States and used the combined patient resources across the different consortia to really demonstrate that a particular gene called DIS3 appears to be related to susceptibility to multiple myeloma. So these people have inherited this particular germline genetic variant, and as a result of that, inheriting that particular variant, you're increasing your lifetime risk of having a multiple myeloma. And this is the first time this has been demonstrated in this particular way, and the idea is that through our international consortium we can firstly identify the particular susceptibility and then see if it is actually remains sufficiently statistically robust across the different populations. And we hope then that can be translated into understanding the susceptibility, but also informing the patient population about their risks and so forth. Okay, so the three key points that we've done in the family working group is that there's a lot of challenges in what we do. When we identify one of the problems we have, or one of the challenges rather we have in the family working group, is we're dealing with really rare events. And I think one of the most key important things is when we deal with these really rare events is to really understand what is a meaningful and biologically relevant result compared with the play of chance. And in fact, that is perhaps one of the biggest challenges in the science field in general. What is true? What is a meaningful observation, and what is just simply the play of chance? And I think one of the most meaningful things we've established in the familiar working group is that we need these type of consortia to be able to really make the distinction between a meaningful result and a chance observation by using our international collaborations to identify a particular interesting finding in one group and then extrapolate it to multiple populations. It's perhaps the most important fact. And there's a bunch of challenges with that. So there's the collaborations, establishing the trust across the research community to make sure that everybody is willing to participate, and I think that's again something that's fantastic with Interlymph, that there is such a vibrant community that's able to work together. It also involves the patient community to be able to provide the biological researchers the commitment to the research, and to be able to collaborate internationally to make these kinds of insights. And at the end of the day, it's, I think, for me at least, it's the only way we can do this type of study is to have a meaningful interaction between the researchers, the patient community, and and also the people that support the research, like the Lymphoma Coalition. I think it's really important that we put them all together through a consortium like Interlymph and make sure that these happen. And of course that takes a lot of effort. There's an enormous working group. The family working group is certainly not just me. It is a bunch of committed researchers that are working towards this particular goal. And also the patients themselves, their commitment to understanding the disease and helping us understand how this works. And we hope also to be able to translate that knowledge back to the patient community. And we hope that this type of research will be fruitful in that goal.